Derived from the anterior abdominal wall, where the testes migrate from their fetal location inside the body to their location outside the abdomen. It is important that the testes undergo this migration because viable sperm will not be produced at internal body temperatures and will develop only at cooler temperatures found within the scrotal sac. Two muscles associated with the scrotum help to regulate the temperature of the testes. The cremaster muscle is formed from skeletal muscle fibers derived from the internal oblique muscle of the abdomen. It reacts to decreasing temperatures by contracting to elevate the testes toward the trunk. The dartos muscle, consisting of smooth muscle fibers, wrinkles the skin of the scrotum in response to cooler temperatures. This action reduces heat loss around the testes. To reveal the testis, the left side of the scrotal sac has been opened. Each testis is about 2.5 centimeters in diameter and 4 centimeters in length. The layers surrounding the testis are derived from structures associated with the abdominal wall. One of these coverings is the tunica vaginalis, which is derived from the peritoneum. Deep to the tunica vaginalis is the tunica albuginea, the dense fibrous capsule of the testis. Inward extensions of the tunica albuginea delineate 200 to 300 individual lobules. Each lobule contains 1 to 4 tightly coiled seminiferous tubules, which are the sites of sperm production. 3 cell types are associated with the seminiferous tubule. In this illustration of a cross-section of a seminiferous tubule, we can identify these 3 cell types. Lying in the interstitium of connective tissue between adjacent tubules are the interstitial endocrinocytes, or cells of Leydig. Their most important secretion is the hormone testosterone.
We saw that.
This hormone plays several different roles in the development and function of male reproductive organs throughout puberty and adulthood. Another cell type that is found inside the seminiferous tubule is the sextentacular, or Sertoli cell. This cell nourishes and supports developing sperm within the tubule. The remaining cells located in the testis are developing sperm, or gametes, which are in various stages of spermatogenesis. Spermatogonia, the stem cells that divide to produce the primary spermatocytes that will undergo meiosis, lie close to the basement membrane of the tubule. As gamete development occurs, primary spermatocytes appear in the middle region of the tubule, and then the more mature secondary spermatocytes are formed somewhat closer to the lumen. The spermatids are located near the lumen, with the immature spermatozoa lining the lumen and ready to move out of the testis. Follicle-stimulating hormone and testosterone are important hormones that regulate spermatogenesis. Each sperm contains 3 regions: a head region, midpiece, and tail. The head is 3 to 5 microns long and contains a nucleus with DNA and an acrosome, which caps the head. The acrosome contains hydrolytic enzymes necessary for penetration of the egg by sperm. The midpiece is 5 to 7 microns long and loaded with mitochondria. These powerhouses produce ATP necessary for movement of the tail, or flagellum. The flagellum is about 45 microns in length. Following the release of immature sperm into the lumen of the seminiferous tubule, the sperm travel through a series of ducts in the testis to reach the epididymis. First they travel through the straight tubule, then the tortuous reedy testis, and finally the efferent ductule to reach the epididymis.
Nancy, we want us to know all of those? No. Probably not efferent ductules, not reedy testis. Good point. And you don't really have to know about the sperm, but it's kind of interesting to hear about acrosome. It helps it to penetrate the membranes and cells around the egg. But I'm not going to test you.
Final maturation of sperm occurs as they travel through the coiled duct within the epididymis. During this process, the sperm learn to swim and finally become capable of fertilizing an egg. The maturation process can take 2 to 3 weeks. The epididymis has 3 portions that will be identified on our specimen. The head is the region where the efferent ductules from the testis join the ductus epididymis. The body contains a highly coiled tube which continues into the tail portion. Sperm travel from the tail of the epididymis to the ductus deferens, which is sometimes referred to as the vas deferens. It is approximately 45 centimeters long and travels in the scrotal sac near the epididymis towards the inguinal canal. The ductus deferens is the structure that is ligated during a vasectomy. The spermatic cord contains structures traveling to or from the testis. These structures within the cord are enclosed in layers of tissue that are derived from the anterior abdominal wall. The spermatic cord travels through an oblique passageway in the inferior border of the abdominal wall called the inguinal canal. The inguinal canal, which has been opened in this specimen, is about 5 centimeters in length. Its floor is formed in part by the inguinal ligament. The abdominal musculature also contributes to the formation of the canal. The left spermatic cord has been opened to reveal one of the most important structures within the spermatic cord, the ductus deferens. Another visible structure within the cord is the pampiniform plexus of veins which surrounds the testicular artery. The relationship of the veins to the artery functions to cool the arterial blood as it flows towards the testes. Other structures found within the cord include nerves and lymphatic vessels. For orientation, we are looking down into the pelvic cavity from a superior view. We will identify the aorta, the inferior vena cava, the common iliac arteries, and the posterior side of the bladder. We can trace the pathway of sperm and the location of 3 accessory sex glands, which secrete fluid that mixes with the sperm to form semen. These fluid secretions provide a transport medium and nutrients for the sperm. The pair of ductus deferens carrying sperm from the testes enter the abdomen via the inguinal canal and travel on each side toward the posterior bladder. As they approach the bladder, each expands to form the ampulla portion of the ductus deferens. The first pair of accessory glands can be seen in this specimen. The seminal vesicles are located behind the urinary bladder near its base. They are about 5 centimeters in length and produce an alkaline secretion that is rich in fructose, ascorbic acid, and a coagulating agent. Secretions from the seminal vesicles produce about 60% of the volume of semen. In this illustration of the posterior aspect of the bladder, we will first identify the ampulla of each ductus deferens. The union of the ampullas and the ducts of the seminal vesicles form the ejaculatory ducts. 2 ejaculatory ducts, one from each side, open into the prostatic portion of the urethra. The single prostate gland forms a donut-shaped ring around the prostatic urethra. Prostate secretions are milky, slightly acidic, and contain proteolytic enzymes which include a liquefying agent. Secretions of the prostate account for about 30% of the total volume of semen. Another pair of accessory glands, the bulbourethral glands, are quite small and are located inferior to the prostate gland within the urogenital diaphragm. The ducts of the bulbourethral glands open into the spongy urethra in the penis. These glands secrete an alkaline fluid prior to ejaculation that protects sperm by lubricating the lining of the urethra and neutralizing its acidic environment. demonstration, we can identify the tissues that form the penis. Most of the penis is erectile tissue. First, we'll identify the urethra, which is surrounded by the corpus spongiosum. The corpus spongiosum attaches to the urogenital diaphragm at the bulb of the penis and distally enlarges to become the glans penis. The opening of the urethra in the glans penis is the external urethral orifice. A cup of loose skin, called the prepuce or foreskin, surrounds the tip of the glans penis. There are also paired dorsal erectile tissues called the corpora cavernosa, or singularly called a corpus cavernosum. The proximal end of each corpus cavernosum is the crus of the penis, which attaches to the ischial and inferior pubic rami. These two crura and the bulb form the root of the penis. The shaft or body extends from the root to the tip of the penis. To get a better understanding of the anatomical relationships of the erectile tissue, we will view a cross-section of the penis. The urethra is surrounded by the corpus spongiosum. The paired corpora cavernosa are situated dorsally. Notice that the corpora cavernosa make up the bulk of the penis. The erectile tissue is highly vascularized and becomes engorged with blood during sexual excitement. On a hemisected specimen, we will review some of the structures of the male reproductive system that we have just described and identify the three portions of the male urethra. The bladder in the male is located posterior to the pubic symphysis and anterior to the rectum. Lying inferior to the bladder is the prostate gland. The male urethra is approximately 20 centimeters in length and functions to carry both urine and semen. The male urethra extends from the bladder through several structures to reach the exterior and contains three named portions. The proximal prostatic urethra courses through the prostate gland. The middle portion of the urethra passes through the urogenital diaphragm and is called the membranous urethra.
Or intermediate part. They've kind of changed it now.
The distal portion extends through the length of the penis as the spongy or penile urethra. The erectile tissues that can be identified in this dissection are the corpus spongiosum and the corpus cavernosum. The bulb of the penis can also be seen in this dissection.